Welcome to the second part of my talk. I will be speaking of ocular proprioception and visual localization and introduce the methods we have for studying ocular proprioception in humans. First of all, I'd like to draw your attention to the fact that visual information is not sufficient to locate visual objects relative to the body. On my slide, you see that the red dot, creates the same image on the eye's retina in both panels. However, here it is situated right in front of the nose, whereas here it is to the right. So retinal information cannot tell us where the dot is relative to the body. Of course, there are rare situations when we can see the nose together with the dot, but those are the exception rather than the rule. So for all practical purposes, extraretinal information is necessary to locate visual objects relative to the body. We have a number of sources that inform the brain about the direction of gaze. And I've listed them here. So they fall broadly into categories: information that tells us about the rotation of the eyes in the head, and information that tells us the rotation of the head on the trunk. So today I will be focusing on extraocular muscle proprioception and a little bit about the corollary discharge. There are two opposite hypotheses about how ocular proprioception and corollary discharge contribute to visual localization. On the one side, some authors suggested that ocular proprioception is used alongside other signals for visual localization. On the other side, other authors hypothesized that ocular proprioception is not necessary and it merely calibrates the corollary discharge over days or weeks. Let me just explain in more detail what the corollary discharge is. So this is a model that was devised for estimating the state of the limbs, not the eye. But it can be used to understand what happens when the brain estimates the rotation of the eye in the orbit. So an ocular motor command is issued by the brain, and a copy of this motor command called corollary discharge is run through a forward model, which is essentially a simulation that estimates where the eye will end up after the movement. Corollary discharge is a prediction, and because it is a prediction, it is available before the eye has moved. On the other hand, the motor command results in a movement of the extraocular muscles, and that causes sensory input, ocular proprioceptive input, which is another source that can be used for estimating the position of the eyes. Ocular proprioception arrives to the brain slower because of the delay in the pathways, in the ascending pathways. However, it is very accurate because it is an actual measurement of the rotation of the eye in the orbit. So the two hypotheses I mentioned in my previous slides are about what happens, how these two sources of eye position information are combined. The first hypothesis that states that ocular proprioception is used for visual localization has been proposed initially by Sherrington more than 100 of years ago. However, a very convincing demonstration of this hypothesis was given by Gauthier and colleagues. They used a very neat way of influencing ocular proprioception, and the scheme of their methods is given here. The human participants wear a scleral lens that is held onto the sclera using vacuum. This is all under local anesthesia. Now, this scleral lens is fixed in a lateral position, meaning that this eye cannot move. And so, while the other eye fixes on the target, there is a discrepancy between the corollary discharge and proprioception because this eye is blocked in an eccentric rotation. This contraption allows to find out how much of this proprioceptive input, of this mismatched proprioceptive input, is taken into consideration when estimating the rotation of the eyes in the orbit. And this can be measured by asking the participant to tell us where a dot is relative to the nose, just as in the slide I presented before. I had the opportunity to try this method myself during an experiment in which we tried to validate the transcranial magnetic stimulation method for interfering with the ocular proprioceptive input. So I tried this myself, and it looks like this. This is with thanks to Dr. Newman and Dr. Knox. It's all under local anesthesia. This is an eye tracker. I am in a chin rest. You can see the scleral lens here with a stalk fixated in a lateral position, and the system that ensures that the lens stays on the eye, so the vacuum system is not as bad as it looks. Now, the model that Gauthier proposed is that the corollary discharge and ocular proprioception are combined together, and they estimated the weight of ocular proprioception to be about 30%, and this was then confirmed by different authors the year after. Now, that's not the only hypothesis. From neurophysiology studies in animals, it was proposed that efferentoscopy, which is another name for the corollary discharge, provides the eye position information required for visually guided reaching. So ocular proprioception is not necessary for this task. These results were obtained after cutting the ophthalmic branch of the trigeminal nerve, which I told you in part 1 brings ocular proprioceptive information to the central nervous system. And they found that after surgery, the monkeys were able to locate visual targets relative to the body to reach to them accurately. There was no reduction in accuracy nor in the and no increase in the standard deviation of reaching. And from here, Lewis and colleagues concluded that proprioception is not necessary and efferentoscopy or corollary discharge sufficient. So using the same scheme that I used before, their model would be that only the predictive, the fast signal is used, and ocular proprioception is not necessary. Steinbach proposed that ocular proprioception is used as a long-term calibrator of the corollary discharge. Steinbach worked with patients with strabismus. So I had the opportunity to work with a patient, RW, who had a focal, very localized, very small lesion in the somatosensory cortex, and you see the arrow that indicates the lesion. RW presented with double vision. The doctors could not tell what the problem was because her vision seemed to be intact. So they conducted an MR scan of the brain and found out this small lesion. This lesion is difficult to see on this slide, so it's here in red, and you see a good overlap with the primary somatosensory cortex, and also some overlap with a proprioceptive representation of both eyes. So that indicates it that this patient might have suffered a stroke in the eye proprioceptive area that could explain her double vision. So this was an excellent opportunity to test the ideas proposed by Gauthier and colleagues and by Lewis and colleagues and see whether this patient would or would not be impaired during visual localization. So the experiment is very similar with what Gauthier and colleagues did. This is complete darkness, again, to avoid any visual cues as to where the targets are relative to the body. And it goes like this. An LED appears and the patient tells us to move the LED to the left or to the right until she feels that the LED is straight in front of her nose. We measured error as the difference between the location of the LED at perceived straight ahead and the actual location of straight ahead. The performance of the patient was compared with 19 elderly healthy volunteers, and we used single subject statistics for the comparison. Now, what would one expect under the first hypothesis? So if proprioception contributes to visual localization, and we assume that in this patient proprioception is not accurate, then we would expect to see an error in visual localization. On the other hand, if Lewis and all are correct and proprioception does not contribute at all to visual localization, then we should see no error at all. And it seems that Lewis and colleagues were right. We saw no error at all in visual localization. Now, of course, our first thought here was, has this patient indeed a problem with ocular proprioception, because we had not confirmed that. And so to do so, we asked her to do the same manipulation as I was mentioning in the first part. So brief push to the eye, then open the eye and conduct the visual localization tasks. So here we introduced a mismatch between ocular proprioception, because we pushed the eye, there's a passive eye movement, and the oculomotor command, which, if ocular proprioception was inaccurate, that should give an effect on visual localization, assuming that proprioception is important for visual localization. And indeed, in this situation, we found an error. So as you can see, the patient was pointing to targets that were more towards the left in the direction of the push than the control. So a large error in the patient, an increase in standard deviation. So This result looks more like the result of Gauthier and colleagues, suggesting that proprioception is important for visual localization. So to conclude, based on these results, I think both models are correct in different situations. So when ocular proprioception and corollary discharge are mismatched, like in the condition where the patient pushed the eye, then opened the eye to conduct the task, then ocular proprioception is used alongside the corollary discharge. Whereas when the ocular proprioception and corollary discharge provide matched information, then corollary discharge is used alone. So the conclusion is that ocular proprioception is continuously monitored to be incorporated into the eye position estimate when a mismatch with corollary discharge is detected. One problem with the methods I presented so far is that they are invasive or accidents of nature, which restricts their usefulness for research. It's simply logistically very difficult to use these methods. A less invasive way to interfere with ocular proprioception in humans is to use inhibitory repetitive transcranial magnetic stimulation over the somatosensory cortex. The rationale for this method comes from an experiment in monkeys that I presented in the first part of my talk. To give you a brief summary again, the monkeys fixate in different directions in the visual space and at different eccentricities, and the researchers recorded from the central sulcus, so from the primary somatosensory cortex. And as you can see here, this neuron, which is sensitive to targets at 120 degrees, fires increasingly with an increasing frequency the more eccentric the target, for instance, like this. Now, from this pattern of activity of a single neuron, one could assume that over the population of neurons, the firing rate would increase the more eccentric the target in all possible directions, and that with transcranial magnetic stimulation, one could bring this activity level down. And so, say for a rotation of 10 degrees, the activity level will no longer be 40 spikes per second, but smaller, 20 spikes per second, which would correspond to a perceived rotation of 5 degrees. So this is why I thought that repetitive transcranial magnetic stimulation in inhibitory mode brings down the neural activity in the oculoproprioceptive area, causing a perceived underestimation of eye rotation. At the somatosensory cortex that receives a projection from the extraocular muscles, we used the fact that in the somatosensory homunculus, the eye is located just below the projection of the hand muscles. And so, because the representation of the hand muscles in the motor cortex is very easily found with transcranial magnetic stimulation, a TMS pulse produces a twitch in the hand muscles, then we place the coil in relation to the motor cortex of the hand. So what we did is to go 3 centimeters posterior and inferior from the primary motor cortex to reach the somatosensory cortex. And we verified using neuronavigation, using structural MRI, that we were indeed in the postcentral gyrus. The choice of the primary motor cortex as a control area is because the primary motor cortex does not control the extraocular muscles, so it's a neutral area. The extraocular muscles are controlled from the frontal eye field, which is further anterior from the primary motor cortex. So this is a good control area. The experiment is designed like this. Transcranial magnetic stimulation in inhibitory mode is given offline. This is a train of TMS pulses whose effect we know outlasts the duration of the stimulation by up to 1 hour. The experiment the task is given twice before and after transcranial magnetic stimulation, and we are looking at differences from the pre-TMS to the post-TMS session. And this is the setup in the Mile lab in Birmingham with my colleague Jonathan Winter assisting. Now, let's go back to the hypothesis that ocular proprioception plays a role in visual localization. We use transcranial magnetic stimulation to test this hypothesis. The task is the same task that I presented before. It goes like this: an LED lights up in complete darkness, and the healthy volunteers say left or right until they feel that the target is straight in front of their nose. And our hypothesis is that after transcranial magnetic stimulation of the somatosensory cortex, the participants will underestimate the rotation of their eyes in the orbit. So, before TMS, a target that is straight in front of the nose will be perceived as being straight ahead. After transcranial magnetic stimulation, a target that is more to the right would be fixated, and because this angle will be underestimated, will be perceived as being smaller, this target would feel as being straight in front of the nose. So we expected to see an error to the left in the post-TMS session compared with a pre-TMS session. And that's exactly what we found, an error about 4 degrees to the left in the post-TMS session relative to the pre-TMS session. And this was specific for the somatosensory cortex and did not occur after the motor cortex stimulation. We had several control conditions in that paper to make sure that this is caused by a change in the proprioceptive input, but this is the experiment that validated the use of transcranial magnetic stimulation as a tool for interfering with ocular proprioception in humans. So, in conclusion, ocular proprioception contributes to visual localization in humans, and transcranial magnetic stimulation over the somatosensory cortex is a valid method for studying the role of this somatosensory modality in the extraocular muscles. And this concludes this part of my lecture.